So the first part over here that you can see, this is called as the eyepiece. So you can see that there is an eyepiece kept inside a container. Okay. So your eyepiece will look something like this. This is the eyepiece. Okay. This is called as the eyepiece. So this will be inserted. You all can see that there are knobs over here. Okay, there is one knob over here and there is one knob. This knob is called as the force adjustment. Okay, this is called as the force adjustment and this one is called as the fine adjustment. Whenever you are lifting a microscope, you will not lift it like this. You will use the handle like this. This is the handle of the microscope. Okay, so what do you call this? This part. You all can see there are three ends. You all see there are this. You all keep our slides on the stage. Okay, and this is called as the mirror. Okay, here you are going to check and adjust the light and then see this light. Okay, you are going to try and focus the light on the mirror. Then you are going to peep inside the microscope and check if you can adjust for the lighting. If you can adjust the lighting over here, then the image that you are trying to view will get visible. Okay, this light gets visible. Okay, here shall we have a quick recap of what we learned? What is this called? Eyepiece. Eyepiece. This is eyepiece. What is this called?
Body view.
Body view is one of the body view. Okay, what model?
Force adjustment.
Force adjustment. This is force adjustment. What model is this? This is
Fine
fine
adjustment.
adjustment. What is this?
Hand
Hand. What is this?
Stage.
Stage. This is the stage.
Right is and this is fine adjustment. This is handle. This is desk. This is stage. And this is adjustment. Force adjustment. Force adjustment.
This is the body.
No, this is body tube.
This is the force adjustment. This is the fine adjustment. This tube that you see, we call this as a body tube. And these are, what are these? These are objective lenses. Okay? Understood? 2.2 to demonstrate osmosis. What we have on the plate over here is the dried raisins. We've got dried raisins on the plate, and in one container what we have done is we have taken the raisins and soaked them in plain water for a couple of hours. After seeing the results, what do we see over here? How is the raisins over here now? It's like that. It is dried. After soaking the raisins, if you pick one raisin and see what has happened to the raisin over here, it has it's become bigger. It has increased in size. So you all can see that what has happened to the raisins. It has increased in size. It is swollen up. That means that from where did the water get inside the raisin? Through the skin. The skin is called the semi-permeable. Skin of the raisin is a semi-permeable. Now the raisins was dry. That means that is there water inside this raisin? How much content of water is there over here? Very less. Very less water is there in this. So when you put it in water, what happened to the raisins? Started absorbing the water. Started absorbing the water. Because of this, the raisins started increasing in size and spreading out. This process is called as osmosis. What is this process called? Osmosis. it depends on the kind of solution that we take. So if the content of water content is same, outside also it is same, inside also it is same, then what will happen? Will osmosis occur? No. If in that case, if the water level outside and water level inside is same, then such kind of solution is called as isotonic solution. What kind of solution is that? Isotonic. Supposing the water content outside is more, inside is less. What kind? It is hypotonic solution. If the water content is less and in the reagents, okay, the water is more, then what kind of solution? Hypertonic solution. So based on these solutions, there will be the exchange of water exchange. Diffusion will happen. Okay, diffusion will happen. When this kind of diffusion occurs, when the water moves from what kind of concentration? Higher